featured slide seeing hidden structures with holographic microscopy. That's it. All right, and you can start whenever you're ready.
Let's say you've got a friend from North Carolina. Your friend comes out for a visit. You go to the Grand Canyon, Wupatki, out to Painted Desert. Your friend thanks you for being a great tour guide and then heads on home. 6 months later, you hear your friend is in the hospital, suffering from valley fever, made worse because it wasn't diagnosed for 2 months after the symptoms first appeared. Here at NAU, in a tiny basement laboratory, we're working on a method to lay the groundwork to understand and diagnose these diseases very quickly. Our method depends upon a principle of physics that allows us to see transparent microscopic objects. And this is important because these diseases present with very non-specific signs, rash, fever, but they have a very specific microscopic signature. All right, if they have a specific microscopic signature, why doesn't that clinician in North Carolina just take a blood sample and look under the microscope? Because biological cells are essentially transparent. It's like trying to see a bunch of glass shards in a glass bowl filled with water. You aren't going to see anything. So standard imaging techniques rely on special sample preparation to add color to those microscopic structures. It's kind of as if each one of those glass shards had a different color inside that bowl. The problem is that special sample preparation is expensive and time-consuming, and no clinician is going to order an expensive, time-consuming test for a rare condition until they eliminate the more likely possibilities. And in North Carolina, valley fever is not very likely. Our technique replaces that special sample preparation with a special imaging method that requires no sample preparation. It's called digital holographic microscopy. And whereas in a regular microscope, light comes up through the slide and you just look at it, in our technique, we take a laser beam and split it into 2 parts. One part goes through the slide and the other goes around. When they're recombined with one another, they create a pattern of darkness and light. We then mathematically process that pattern of darkness and light to reveal those hidden microscopic transparent structures. We've already proven the principle. Now we're working on adding a new capability, combining multiple images together to create a 3D model of the cell. For now, it's a research capability, but eventually it could serve as the foundation for a method to quickly, accurately, and inexpensively diagnose difficult to detect diseases. Thank you.